So today we're talking soil sampling out here at Stanley, and we're going to give you a demonstration of how to take a soil sample for a Salina soil biome test. We also would recommend that you look at production areas, so you might be testing your best paddock and your worst paddock, or you might be looking at two different soil types, but basically blocking down your testing into productive areas. Select the location. Here we've got a row of trees that these trees are actually not irrigated, but either way you would pick an area in line with the root zone. If you're in an irrigated farm, you'd be taking your soil sample from under the drip line. Basically just scrape away the top couple of centimetres, and what we're looking to do is we're looking to take a sample from the first 10 to 15 centimetres or 6 to 7 inches of your soil microbiome. Something like that. In your soil sample kit you'll find two types of vials. The brown cap vial with the Salina branding on it and the QR code. This is the main vial that contains the soil buffers that will be sent to Salina for testing. The second vial is a grey top vial, just a generic vial. We keep these vials at Great Southern Biology as backups. You'll also receive a set of rubber gloves in the soil test pack. It's important that you want to keep your own DNA out of the sample. You're not testing your own DNA, you're trying to test the soil DNA. Today we've brought a little measuring jug, but ordinarily you could use a bucket or any sort of receptacle. And then just from the wedge of soil you've taken, you're just breaking that up a little bit and taking a sample from it. So something like that. You'll end up with around about that much from each location. Break them up a little bit, and we'll mix 6, 7, however many of these we want to, anywhere up to about 10 or 12 of these types of sub-samples together to make a bigger sample, and then we'll select from that to fill the vials that we were discussing earlier. So we'll just move up to our next location, pick another spot between a couple of trees up here a little bit. Once again, we're not trying to scrape the top soil off, we're just removing the debris. So we're looking at that top 6 to 8 inches. So just get ourselves a nice sample there, and a handful or two, and we're off to our next sample. Alright, so we've been through the orchard and we've picked up our, I think we ended up getting about 8 samples in total, and now we're going to take our final sample from our sub-samples. So using a relatively clean bucket, just tip your samples in. So our soil, that's all our combination of soil samples. Give those a really good mix up, try and get them as homogeneous as you possibly can. We've got our 2 vials. Our Salina vial with the brown lid, which is the predominant vial that we're focusing on here today. This is the vial with the buffers in it that the soil sample will go to the laboratory in, and then we're going to take a sample out of our bucket and fill both of these tubes. So we'll actually start with the backup vial in this case. Try and keep the leaf and root matter out of it best you can. And then the A vial with the Salina branding we spoke about before. So there's your little spoon for filling. That's about right. You can see there those buffers are just starting to absorb through the soil. By the time you get back to the car, they'll be absorbed all the way through. They'll preserve that soil sample all the way until it gets to the lab for testing. So there's the job done. 2 vials filled up, ready to go. Brown top vial for the lab, grey top vial to come back to GSB for storage. Put both of those in the envelope or the box rather, that we've provided you with. send it back to Great Southern Biology and we'll handle the rest from there.